Before he reached for the cup of hemlock, which he drank to end his life, Socrates despaired of the way the natural sciences were fixated on mechanical, material explanations of causation. The gene replication translation models of early molecular biological information processing demonstrate how life took over the quantum mechanical causation of the physical world. Molecular biology encompasses both chemical and computational modes of description and analysis. Figure A shows protein synthesis as the chemical concatenation of amino acids at the right head of the ribosome. Figure B shows a computational lookup table, the genetic code, which assigns a unique amino acid type to each nucleotide triplet codon encountered by the lower read head. Translation represents a mapping from the molecular sequence information held in genes onto the catalytic functions of protein catalysts. In this schematic representation of the process, a set of genes G is translated to produce proteins P, using the rules of the genetic code. However, there is another important mechanism of biological information processing. What is life? Schrödinger envisaged genetic material as an aperiodic crystal that contained a code script. Davies and Kuppers emphasize that information is central to molecular biological processes. The current paper by Wills suggests that life was created when computational processes took over self-organizing material processes. Molecular self-organization. Schrödinger described how the ordered configuration of molecular biological processes are maintained by dissipating energy. Prigogine showed how systems can undergo self-organization to form dynamic dissipative structures of reduced internal entropy. Turing showed that spatial patterns can emerge spontaneously from homogeneous solutions due to the coupling of chemical reactions and molecular diffusion. Self-organizing autocatalysis. The molecule being copied, A, serves as a template which assists in the formation of the new copy, without itself being changed by the reaction. The autocatalytic feedback feature of the replication is shown explicitly in C. The replication of any polymeric sequence produces a variety of offspring containing 0, 1, 2, etc. random mutations. Below a certain threshold in the replication error rate, a population of individual replicating polymers evolves into a self-organized population called a quasi-species. Template-directed nucleic acid replication is usually catalyzed by a specific replicase enzyme R. The specificity and accuracy of a replicase determine the rate of replication of individual polymer sequences, guiding the self-organizing process of Darwinian selection, survival of the fittest polymer molecules. Now we consider the translation of some very special proteins, a coding set of assignment catalysts T. The set T is chemically autocatalytic. Production of the set T requires T. Each individual assignment catalyst's Ti is functionally equivalent to an AARS enzyme that attaches a specific amino acid to a cognate tRNA, according to the strict matching rules of the genetic code. Translating the genetic sequences G according to the set of codon to amino acid assignments catalyzed by T produces proteins with the sequences of T. So we say that the mapping from G to T is computationally reflexive. Reflexive information G guides the self-organization of the coding set T of assignment catalysts. In a complete GRT system, a replicase enzyme is produced by translation of an appropriate gene. The replicase R guides the self-organizing selection of the reflexive information G needed to produce the set T of coding enzymes. Gene replication translation models demonstrate the simultaneous, mutually guided, computational self-organization of both translation and replication. The molecular mechanism is Turing-like reaction-diffusion coupling. The minimal computational condition for molecular biological self-construction is the simultaneous, local occurrence of the assignment catalysts T for a binary code and corresponding reflexive genetic information G, shown here as the separate, complementary, anti-parallel sequences of a double-stranded nucleic acid. The self-organizing transition to molecular biological coding consisted of mutually guided symmetry-breaking bifurcations in the extant gene and protein populations. This transition is from an effectively random distribution of both polymer types to separated pairs of matched quasi-species, corresponding to two ARS-like enzymes, T, that execute a binary code, along with reflexive genetic information G, which encodes them. Each pair of code-expanding quasi-species bifurcations potentiates the next, enabling stepwise refinements in the genetic specification of protein encoding. Such specification is mediated by the controlled deportment of amino acids with diverse physico-chemical properties to produce folded proteins with increasingly refined functionalities. The evolution of the ARS enzymes records the reflexive harnessing of diverse amino acid properties to construct, in terms of nucleotide triplet codons, an information-based, coded operational map of those very same physico-chemical properties of amino acids. Genetic coding took control of events in the physical world by bootstrapping and embedding its own system of semantics into molecular processes. Conclusion. The advent of genetic coding consisted of the mutual guidance of two self-organizing processes, the replication and translation of genetic information, to form a combined self-guided, physically constructive computational system, the evolution and refinement of which transcends simple physical causation. Do you want to show your research progress to more scholars? Contribute an entry that will be linked to your paper on the encyclopedia platform, providing an opportunity for over 35,000 scholars to access and explore your research. Have you ever tried to show your latest research in a vivid way? Encyclopedia SciPlayer provides a service with which you can transfer your research to a live video format. Do you want to publish a new paper? Encyclopedia has an international, peer-reviewed, and open-access journal. Do you require a discount when you publish research in journals? Anyone who contributes to Encyclopedia can obtain vouchers for MDPI Journal's article processing charges. Encyclopedia aims to create an open-access knowledge-sharing platform for all scholars. To find more information, search for encyclopedia.pub.